Good morning. Welcome to the Medicare Advisory Panel on Clinical Diagnostic Laboratory Test meeting. My name is Dr. Rashida Arthur, and as a member of the Medicare Clinical Laboratory Fee Schedule Policy Team and the Division of Ambulatory Services, I am the designated federal officer for the CDLT panel. I would like to begin by expressing my appreciation for those who were able to participate, either in person or via the webinar. We'd also like to extend a warm welcome to members of the Medicare Advisory Panel on Clinical Diagnostic Laboratory Test. This meeting allows an opportunity for the CDLT panel to provide input and recommendations to the Secretary and the Administrator of CMS on the appropriate basis for establishing payment amounts for new or substantially revised healthcare common procedure coding system codes being considered for Medicare payment under the CLFS for the next calendar year. This meeting also provides a forum for the public to respond to direct questions posed by the CDLT panel members in a real-time manner. Please note that we will be discussing only the codes addressed in our Clinical Laboratory Fee Schedule CY 2027 Test Codes Update file, which is found on our CLFS Annual Laboratory Meeting webpage. Comments and recommendations on whether codes identified as gap fill codes from last year's final determination is a separate and distinct process that will not be discussed at this meeting. The end of the Protecting Access to Medicare Act data reporting period ends on July 31st, and we'd like to remind applicable laboratories to please submit your applicable information by that deadline. Please refer to the CMS website for more details. This year, we will hold a public meeting that includes the CDLT panel on Tuesday, September 15th, 2026, and Wednesday, September 16th, 2026. During that meeting, the public will have an opportunity to present recommendations on the appropriate basis for establishing payment amounts for CDLTs for which CMS received no applicable information during the data reporting period from May 1st to July 31st, 2026. After the public provides this input, the panel will provide its recommendations to the Secretary and the Administrator on payment recommendations for those CDLTs. After consideration of the information from the ALM, the public comments, and the CDLT panel recommendations, we generally post preliminary payment determinations on our website in September or early October of each year. We accept additional public comments following the posting of the preliminary payment determinations through October of each year. Unlike a new crosswalk test, the payment amount for a new gap fill test is not established when we determine the basis for payment because it takes approximately 9 months from the CLFS annual public meeting for MACs to establish MAC-specific amounts and report those amounts to CMS. In November of each year, we generally finalize the basis of payment for new and substantially revised test codes and the amount of payment through the annual CMS instruction implementing the updated CLFS for the next calendar year. The public has an additional 60 days from the date we issue our annual instruction on the final payment determination to request reconsideration of either the basis of payment or the amount of payment for new or substantially revised test codes. The public may comment on these reconsideration requests at the next CLFS annual public meeting. Once a code or a lab test has undergone a reconsideration cycle and CMS has made second final determinations on the code or the test, the payment methodology is final. For any new test code that will be gap filled, we ask our MAC or Medicare contractors to develop a MAC-specific gap fill amount by April 30th of the following year. The amounts will then be finalized by September 30th of the year. Unlike a test crosswalk to a CLFS lab test code, the payment amount for a gap fill test is not established when we determine the basis for payment because it takes approximately 9 months for our contracts to establish carrier-specific amounts. After prices for gap fill codes are developed by the Medicare contractors, we will post them on our website and accept comments for 60 days. Later in the year, when the gap fill payment amounts are posted on the website as final, we will accept reconsideration requests on the gap fill payment amounts for 30 days. Once the reconsideration process is complete for the cycle, the determination is final and would not be subject to further consideration. Lastly, I'd like to share a few housekeeping issues or items. Please note the following. As a reminder, the agenda and the code list may be found on the CMS website. Standby speakers, if you are not speaking, please remember to keep the line muted. Regarding input from standby speakers, we will seek input only when a CDLT panel member has a specific question. That is, standby speakers cannot address the chair or the panel members directly. They can only do so when called on for a specific question. Another important housekeeping rule is to honor warnings when the discussion or a given speaker is potentially off topic. When the discussion begins to deviate from the specific question posed, the panel chair can intervene to reorient the discussion and guide standby speakers back towards the original question. Additionally, like last year's meeting, if during the discussion of a code, a new recommendation is proposed, that discussion for the specific code will be concluded and we will move the discussion to the end of the meeting. This will be done to allow our panel additional time to adequately consider the new recommendation. We'll keep a list of the codes that need to be moved toward the end of the agenda as a discussion of codes occurs. So again, we hope to have a very efficient meeting and we look forward to interacting and hearing from the FACA panel as well as the standby speakers. If you have any additional questions during this meeting, please do not hesitate to reach out to members of the Division of Ambulatory Services team. If you have follow-up questions or issues after this meeting, please submit all inquiries to the CDLT panel resource box. Before I turn the meeting over to our panel chair and meeting facilitator, Dr. Megan Landsberg, I would like to introduce the management team of the Division of Ambulatory Services, Sarah Shiriloso, the director, and Michelle Cruz, the deputy director. This concludes my introductory and housekeeping comments. We will now hear a few remarks from Dr. Landsberg.
Great. Thank you, Dr. Arthur. Good morning, everyone. My name is Dr. Megan Landsberg, and as the panel chair, I will be facilitating today's meeting. Again, welcome to the Medicare Advisory Panel on Clinical Diagnostic Laboratory Tests meeting, where we will seek information and input from our FACA panel. I would like to begin by performing a quick roll call of our panel members. So for each member, when you hear your name, please acknowledge that you're present. Vicki Piselsky.
Present.
Dr. Pranal Chandra.
Present.
Jerry Garner.
Present.
Dr. Chris Chung.
Present.
Dr. Jillian Hooker. Jillian. All right. Jen Jiang, Dr. Jen Jiang.
Yeah, I'm here, present.
Dr. Joe Leonards.
Hello, I'm here.
Dr. Mark Rumpler.
Present.
Dr. Christine Schmatzer.
Present.
Dr. Michelle Schumacher.
Present.
Heather Schappel.
Present.
Dr. Lena Bridges.
Present.
And Jillian. All right, we'll check on Jillian. Now that we've completed roll call, I can confirm we have a quorum. I would also like to briefly note that in addition to the names of the panel members, you can see on the screen the source of the nomination for each member. At this time, I'd like to brief the public on the advisory panel's process for reviewing and categorizing the codes on the list for each year. Over the years, we've created subcommittees to review the list of codes. The purpose of the subcommittee is to maximize the utility of panel member expertise while streamlining the efficiency of the discussion during the meeting. We have created two subcommittees for this purpose. One is the Molecular Pathology Genomic Sequencing Subcommittee, or the MOG subcommittee. And the second is the Subcommittee on Chemistry, Hematology, Immunology, and Microbiology, or the CHIM subcommittee. Each subcommittee has co-chairs. The co-chairs for the MOG subcommittee are Drs. Joe Leonards and Dr. Pranal Chandra, and the co-chairs for the CHIM subcommittee are Drs. Vicki Piselsky and Mark Rumpler. So again, the total number of codes were divided amongst the two subcommittees. There were discussions on how to approach crosswalks and or gap fill for each of the codes within each subcommittee after leveraging panel expertise, considering public comments, utilizing other pieces of supporting information to understand the complexity of each test. And then finally, it's important to understand that the subcommittee or an individual panel member does not make the sole recommendation for a test to CMS. Rather, the full panel makes recommendations on each code when they vote on their choices for either a crosswalk or a gap fill, which will occur during this meeting. Please note that as the panel chair, I will not be voting. Now, we have taken a great number of codes on our list and broken them up into various categories. In general, we try to maintain consistency with the categories that AMA CPT uses in their manuals, and then we look at the type of lab test, the specimen, the analyte, etc. All of this goes into the decision-making process in terms of taking a large list of codes, like the genomic sequencing codes, the microbiology and immunology codes. Furthermore, the panel also looks at the types of specimens, analytes, methodologies, resources, and the indication of the test, to name a few. So again, the whole process of creating subcategories out of the major categories is so that you would recognize when you looked at the AMA CPT book to help promote efficiency during the meeting and to help panel members quickly begin to sort through similar. You will notice that for today, we're going to cover many codes across multiple different categories. So please follow along using the code list provided in Appendix 3 of the agenda. We will begin with the first code, 8XX30, which will be discussed by the CAM subcommittee. So
Yes, thank you. So
Go
the
ahead.
first test on the CDLT meeting agenda is 8XX30, glial fibrillary acidic protein, or GFAP. This test has not yet received a final CPT code assignment. 3 separate associations sponsored this test during the annual lab meeting. 2 recommended a crosswalk to 0548U, which has a similar code descriptor. Additionally, another association recommended an alternative crosswalk to 83884 neurofilament light chain, suggesting that it uses similar resources and supports the same CLFS rate as 0548U. The SIB committee researched the publicly available information for the suggested crosswalks and agrees that 0548U is the most suitable since both represent the same biomarker with similar descriptors.
Do we have any other options for the panel to consider? And do we have any questions or comments from the panel or from CMS about this code? All right, if there are no additional comments or questions, panel members, please cast your vote now. We have our votes coming in.
They are coming in. I think we're waiting for one more. Just bear with us 1 second while we get through this first code. Okay, Dr. Bridges, could you double check that your vote went through?
I am trying to find the link to the polling link.
Okay. I'll send it in the chat to you. Everybody, if you could just give us one quick second. There's always a few bumps for the first one. We will figure it out.
I'm used to seeing it appear as a QR code. And it's not letting me log in for some reason. I don't see the link yet.
Okay, I just sent it to your email.
Okay. Okay.
We
Okay.
got it. That's perfect. Thank you so much.
Sorry.
All right. Locking that. Okay, Dr. Landsberg, back to you.
All right, thank you. All right, so next code is X300U with a final code of 0698U. Jerry, this one's you as well.
Yes, thank you. So X300U is now PLA code 0698U, Traumatic Brain Injury Panel, including GFAP and UCHL1. This essay was sponsored by 2 associations and 1 manufacturer, suggesting a crosswalk to 0570U with a similar code descriptor. The subcommittee researched this test and found that these are, in fact, the same test and agree with the recommendation to crosswalk to 0570U.
All right, are there any new options for the panel to consider? And are there any questions or comments from the panel or CMS on this code? All right, well, if there are no additional comments or questions, panel members, please cast your votes now.
All right, that was everyone. Thank you.
Fabulous. All right, our next code, I'm gonna scroll up a bit. The next code is 0609U. And
Yes.
that's you as well, Jerry.
Thank you. Yes, 0609U. This is a 2 biomarker MAAA blood test for prostate cancer, including total PSA and free PSA with an algorithm. This test appears to be similar to 0550U, which was deleted effective January 1st, 2026. Both 0550U and 0609U have the same PLA code owner. The CDLT panel voted to gap fill 0550U in 2025. After review, the CDLT panel determined that the requested crosswalk from the test sponsor is not appropriate and differs in the number of analytes in the specific isoforms. The assay matches specimen type and clinical use, but does not match on the number of components. On a matter of precedent, the subcommittee researched 0359U, which was presented on June 22nd, 2023, during the annual lab meeting, which outlined 4 markers matching its surrogate of 81539. Therefore, it is recommended that 0609U be placed for gap fill.
Thank you. Are there any other options for the panel to consider for this code? And any questions or comments from the panel or CMS? All right, if not, panelists, please cast your votes.
Thank you.
All right, that all votes are in?
Yes, indeed.
Fabulous, thank you. All right, we'll move on to the next code. So the next code up is 0650U. And Dr. Bridges will be discussing this code.
Yeah, I apologize. I have to be off camera. I actually tested positive for influenza A yesterday, and so I'm very sick. So this code refers to a targeted multi-gene pharmacogenomic genotyping of 9 genes. 81418 is a category 1 multi-gene pharmacogenomics panel. That includes about 6 genes, matching the methodology and resource use. The recommendation is to crosswalk to 81418. An alternative is 0349U, which is a RightMed 27 gene next-gen sequencing pharmacogenomics alternative on the ballot.
Right, do we have any other options for the panel to consider?
A gap fill or to abstain.
And are there any questions or comments from other panel members or from CMS about this code? All right, if there are no additional comments, panel members, please cast your votes.
Okay, thank you.
All right, the next code up is 0652U, and Dr. Bridges will be presenting this one as well.
Yep. This one has the same targeted multi-gene pharmacogenomics genotyping methodology as 0650U with 13 genes, and we recommend crosswalking to 81418 category 1 pharmacogenomics panel that has 6 genes and is size appropriate and keeps pricing consistent with 0650U. The gene list is undisclosed, and an alternative, again, similar to the prior code, is 0349U. So my recommendations are crosswalk to 81418, crosswalk to 0349U, gap fill or abstain.
Thank you. And are there any new options for the panel to consider? Or any other questions or comments from panel members or from CMS? If there are no additional comments or questions, panel members, please cast your votes now.
Okay, thank you.
All right, we're going to switch gears a little bit and we're going to now discuss category 1 code 87638. And I wasn't sure, we've got Dr. Baselski and Dr. Schoonmaker discussing this one.
Yes, thank you. This is Dr. Schoonmaker. I'll be discussing items 6 through 9 and 12 through 17, and then Dr. Baselski will discuss the remainder of the codes later in the day. So looking at 87638, this is a new code for PCR for rubella, the measles virus. We did not have a sponsor recommendation. However, both ACLA and ASM recommended a crosswalk to 87635, which is for the SARS-CoV PCR assay. And we agreed with that recommendation, as both are a single target detection of an enveloped negative sense virus and an emerging pathogen. So our recommendation was to crosswalk to 87635.
All right, and are there any new options for the panel to consider? Or any questions or comments from panel members or CMS? All right, if not, panel members, please cast your votes.
We are waiting on one more vote. If you can check that your selection went through, please. Thank you.
All right, next up, we have another category 1 code, 87183.
Yes, 87183 is a code that is being reconsidered from last year. It is for susceptibility studies, antimicrobial agent, carbapenem resistance genes by PCR per isolate. Last year, the sponsor had recommended a crosswalk to 87505, which is the GI panel of 3 to 5 targets. And the test was finalized as a crosswalk to 87150. With a rationale that all 5 targets may not be run all the time. After discussion at the ALM last month, the sponsor is asking for a gap fill for this code, and multiple specialty societies agree. Upon further evaluation, this test is a multiplex assay evaluating all the targets at the same time on each isolate tested, and therefore the committee recommends that we crosswalk to 87505, which also uses PCR to evaluate 3 to 5 targets per specimen and also addresses the economies of doing the multiplex test. So our final recommendation is to crosswalk to 87505.
Thank you. And are there any new options for the panel to consider? Or any questions or comments from the panel or CMS on this code? All right, if there are no additional comments or questions, panel members, please cast your votes.
Okay, thank you.
All right, so sticking with category 1 codes, next up we have 8XX32.
8XX32 is a new code for a PCR assay detecting multiple Candida species with a call out for Candida glabrata and Trichomonas. The sponsor recommended a crosswalk to 0455U, 2 professional societies agreed with the sponsor, and 2 others suggested 87631, which is a 3 to 5 target assay. The subcommittee, after discussion, agreed with the sponsor and is recommending a crosswalk to 0455U on the basis that both tests are for the same clinical indication, which is STI detection, report the same number of targets, and use the same methodology.
Thank you. Are there any new options for the panels to consider, or any questions or comments from panel or CMS? All right. If not, panel members, please cast your votes.
Dr. Booker, is that you? Hooker, is that you?
Pardon me? Yes.
You're here.
Yes.
Fantastic. Welcome.
Just
All right. All of
messaged.
a sudden we had extra votes.
So sorry.
Perfect. No, you're good. All right. All the votes are in. Thank you.
All right. So next up, we have, just making sure here, 87XX2.
Yes, 87XX2 is for a single target PCR assay for macrolide resistance. We did not have any recommendation from a sponsor. However, 2 professional societies recommended a crosswalk to 87500, and 2 recommended gap fill. We agreed with a crosswalk to 87500, as both of these codes are for the single target of for detection of a single target resistant gene by PCR. So our recommendation is to crosswalk to 87500.
Thank you, Michelle. Are there any new options for the panel to consider, or questions or comments from the panel members or CMS? All right, if not, panel members, please cast your votes.
Okay, we're good. Thank you.
All right, switching gears here. We have next up is code 0635U, and I believe Dr. Rumpler will be presenting this one.
Yes, thank you. So 0635U is a non-invasive skin surface scraping test that uses next-gen sequencing to profile 487 genes via mRNA expression. The panel recommendation is a crosswalk to CP20258U. Both tests use NGS-based mRNA gene expression profiling from a non-invasive skin surface specimen to generate an algorithm-reported likelihood of response to systemic therapy for an inflammatory skin disease.
Fantastic. And are there any other options or questions or comments from the panel members or CMS? No? All right. If not, panel members, please cast your votes.
Okay, thank you.
Fabulous. All right, next up we have 0605U, and Dr. Chandra will be taking this one.
Thank you. And so I did evaluate this code. And before I get into my recommendation, I just wanted to say a quick thanks to all of the presenters who actually did come forth and give some really great presentations and recommendations. That's very helpful to the panel members. And so I just wanted to acknowledge that and appreciate the effort. So this is a code for TPASAB1. It is a digital PCR test, and there's an algorithmic based result of positive or negative. I agree with the sponsor's recommendation to gap fill this code, and that is my recommendation.
Fantastic. And do we have any other options, any questions from panel members or from CMS about this code? No? All right. If not, panel members, please cast your votes.
Okay, thank you.
Fabulous. Okay, back to micro. We have code 0580U, and Michelle will be taking this one as well.
Yes, thank you. Items number 12 to 17 in the next set address the identification of different pathogens by detection of a varying number of recombinant proteins for either IgG or IgM antibodies by immunoassay. The sponsors had recommended gap fill for all of these codes in this set. For the first one, 0580U is a reconsidered code. Last year, the code was crosswalked to 0042U, the detection of Borrelia of 12 recombinant protein groups by immunoblot for IgG. After discussion, the committee agreed that this was the appropriate crosswalk, but we felt that a multiplier of ×2 was appropriate to reflect the added resources to detect 24 recombinant protein targets compared to 12 described by 0042U. And therefore, our final recommendation is to crosswalk 0580U to 0042U ×2.
Fabulous. Are there any other questions, comments from the panel or CMS on this set of codes? No? All right. So panel members, please cast your votes.
Thank you.
All right. The next up is that similar code 0615U.
Yes, this code is for the detection of the Borrelia Lyme disease pathogen, antibody detection of 26 recombinant protein groups by immunoassay for IgM. The sponsor had recommended gap fill. And last year we did not have any information on, 0580U, which was the IgG version. And CMS had crosswalked that to 0042U. For this particular test, we felt that the appropriate crosswalk was to 0041U, which is the Borrelia assay of 5 recombinant protein groups by immunoblot for IgM. It matches the organism and the methodology, and we further recommend that there be a multiplier ×5 to reflect the difference in the number of targets for 0041U versus the 0615U. So our final recommendation is to crosswalk to 0041U ×5.
Fabulous. Are there any other options, questions or comments from panelists or from CMS about this code? All right. If not, panelists, please cast your votes.
We're just waiting on one more response. Dr. Leonards, can you check that your response went through, please? Okay, thank you.
Fabulous. And we're going to keep it rolling with Dr. Schoonmaker and 0636U.
Thank you. This assay is for babsiosis, antibody detection of 20 recombinant protein groups by immunoassay for IgG. We recommend that this be crosswalked to the Lyme disease assay for 0042U with the multiplier ×2 to reflect the higher number of targets in the new test.
All right. And do we have any questions or comments from the panelists or CMS on this code? No? All right. Everybody, please cast your votes.
Okay, thank you.
All right. And I think maybe, Michelle, we might be able to do the next three all in a group since they are similar. 0637U, 0638U, and 0639U.
Yes, they're all for a single condition, pathogen, antibody detection by varying numbers of recombinant protein groups. Two are 0637U and 0639U are for detection of IgM protein groups, while 0638U is for detection of IgG protein groups. And so the crosswalk rationale remains the same, to crosswalk the IgG codes to so 0637U, we recommend to crosswalk to 0041U with a multiplier of 4 to reflect the difference in the number of targets. That is an IgM test. 0638U, we recommend crosswalk to 0042U times 3, again, to reflect the different number of targets. And then 0639U, we request a crosswalk to 0041U times 5, again, reflecting the difference in the number of targets.
Fabulous. Thank you. Do we have any questions or comments about this group of codes from the panel or CMS? No. All right. So I think we can vote on them one at a time. So 0637U, panelists, please cast your votes.
Thank you.
All right. Next code 0638U. Panelists, please cast your votes.
Thank you.
All right, and 0639U, panelists, please cast your votes.
Thank you.
All right, so we're going to switch gears back to molecular again. The next code up is previously code X258U with a final code of 0687U. And Dr. Chandra, this one's yours.
Thank you. So this is a comparative analysis using STR markers, DNA-based assay. The sponsor gave a really nice presentation with their crosswalk recommendation and their rationale, which I completely agree with. And so my recommendation is to crosswalk to 81265 plus 81266 times 0.5.
Thank you. Are there any other options from the panelists? Questions or comments from panelists or from CMS? All right. If not, panelists, please cast your votes.
Thank you.
All right. We are staying with Purnell here for the next code, formerly known as X274U with a final code of 0695U.
Thank you, Megan. This is an oncology code that is a cytogenomic genome-wide analysis. The sponsor gave another great presentation with their recommendations, which was a crosswalk to 81349. That was well done. And I agree with that crosswalk recommendation of 81349.
Thank you. Do we have any other options, questions, or comments from panel members or CMS? All right. If not, everybody please cast your votes.
Thank you.
Thank you. And the next one was a reconsideration of code 0575U, staying with Purnell here.
Thank you, Megan. This is a transplant code. It's a messenger RNA-based gene expression profiling assay. And again, the sponsor gave a very nice presentation with their rationale to crosswalk to 81595, which seems appropriate and commensurate with the resources needed to perform the assay. And so that's my recommendation, to crosswalk to 81595.
Fantastic. Thank you. Do we have any other questions or comments? Panelists or from CMS? No? All right. If not, panelists, please cast your votes.
Thank you.
All right. The next code up, formerly X252U. There we go. With a final code of 0662U. And Dr. Leonards will be taking this one.
Thank you. Just like Dr. Chandra presented, this is a transplantation code. The number of genes and by effort is also matching the prior rationale, and the recommendation by the subcommittee and myself is to crosswalk to 81595.
Thank you. Do we have any questions, comments from the panel or CMS? No? All right. Thank you, everyone. Please cast your votes.
Thank you.
All right, we are staying with transplant here. Code number X257U, new code number 0664U. And I believe Joe is taking this one as well.
Yes, I'm presenting also Dr. Rampler here. We both assessed this code and the next one together. Briefly, this is also RNA expression profiling, transcriptome analysis, NGS, and a analyte algorithmic analysis. This assay is done by the same lab, same NGS workflow, and same specimen type as a prior code, and this test is 0320U, and that's the recommendation by the MOG subcommittee and myself and Dr. Rumpler to crosswalk to 0320U.
Thank you. Do we have any questions or comments from panel members or CMS on this code? All right, if no questions, panel members, please cast your votes.
Thank you.
All right, next code up is 0630U, and we will be sticking with Joe on this one.
Thanks again. Also, recommendation by myself and Dr. Rumpler as well as the subcommittee. After review of the existing codes, there were two that were considered based on the same laboratory, the same microarray platform, and a comparable gene content. We thought that the MAMA print is the most or the closest similar, and therefore the subcommittee recommends two options, but favors the crosswalk to 81521. And as an alternative, we also considered the crosswalk to 81540. That was, however, not quite as well matching. So final recommendation by the subcommittee, represented by Dr. Rumpler and myself, is crosswalk to 81521.
Thank you. Do we have any other questions or comments from other panel members or from CMS? Right. If not, panel members, please cast your votes.
Thank you.
All right, next up, we have X282U with a final code number of 0668, and Dr. Leonards will be presenting this one as well.
Perfect. Yes, this is also an oncology code or an assay in the oncology family for pancreas. The interesting thing here is that in addition to a 5-hydroxymethylcytosine enrichment, there's also a glycan biomarker analysis. And after review of the close similar codes, the subcommittee recommends a combination. First, a crosswalk to 0410U, which is a similar main assay, plus a crosswalk to 86301, which is an immunoassay for tumor antigen detection. And that combination is the recommendation by the subcommittee as well as myself. So that's crosswalk to 0410U plus 86301.
Fantastic. Do we have any other suggestions, questions, or comments from panel members or CMS? All right, if not, please cast your votes, panel members.
Thank you.
All right. Thank you, Dr. Leonards. Next code up is 0634U, and Dr. Bridges will be presenting this one.
Yes, this is also for a breast cancer. It's a cell-free DNA evaluation of 11 ESR1 variants using droplet digital PCR, and it's reported as positive and negative. There's no existing code that shares this droplet DNA PCR methodology and resource profile. So the recommendation was to use a gap fill versus an alternative is a crosswalk to 0332U, which is a pan-tumor epigenetic qPCR for immunotherapy response, but that differs in method, specimen, and intent, but that would be an alternative to a gap fill.
Thank you. Do we have any questions or comments from other panelists or CMS on this code? All right, if not, panelists, please cast your votes.
Thank you.
All right, next up, we have a couple of biomarker codes. So formal code number X251U, final code 0660U.
Yes, I have that code. So this is a multiplex droplet digital PCR of cell-free HPV 18, 31, 32, and 35 in the blood. There's no existing code that shares this HPV DD PCR methodology. The recommendation is gap fill. The alternative is crosswalking to a fraction of the NGS-based 0470U, which doesn't reflect the droplet digital PCR resource profile, but remains a valid alternative in this case.
Thank you. Does anybody have any questions or comments from panelists or from CMS about this code? All right, if not, panelists, please vote.
Thank you.
All right, Alina, I think this one is yours as well. Code
Yes.
number X283U, final code 0661U.
Yes, this code is related to the prior code, but it's a single target droplet digital PCR of cell-free HPV 16 in the blood. Again, there's no existing code that shares the HPV ddPCR methodology, and a fraction of the NGS-based 0470U does not reflect the ddPCR resource profile, but is a valid alternative to the proposed gap fill.
Fabulous, thank you. Any questions or comments from the panelists or from CMS? All right, if not, panelists, please vote.
Thank you.
All right, we're going to switch gears and discuss some chemistry codes here. So next up, we have code number 0604U, and I think Dr. Chong is taking this.
Yeah, thank you. So this is a test for 4 bradykinins using LC mass tandem mass spec. None of the existing codes take into account the pre-analytics that is required to perform this test. So the subcommittee recommends a gap fill.
Thank you. Does anybody have any questions or comments? Many of the panelists or CMS on this code? All right, panelists, please vote.
Thank you.
All right, next up, we have 0614U, and I believe Dr. Rumpler will be taking this one.
Yes, thank you. Notably, 0614U was evaluated without a dossier submitted to this panel by the sponsor. 0614U is a test for primary mitochondrial disease that separates and stains 4 mitochondrial respiratory chain enzyme complexes into a single gel. Uses tissue and produces a qualitative diagnostic result. In that case, the panel recommendation is a gap fill because there's no existing CPT or PLA code that captures a gel-based multiplex assay that simultaneously separates and stains 4 mitochondrial enzyme complexes.
Thank you. Do we have any questions or comments from the panel or from CMS about this code? All right, if not, panelists, please cast your votes.
Thank you.
All right, next up, we have 0654U, and I believe Jerry is taking these.
Yes, this is a test for inborn metabolic error of one enzyme by Western blot using cultured skin fibroblasts. This test had no sponsor for the annual lab meeting and no presentation was received. After researching the assay, the subcommittee found that the test belongs to Children's Colorado Hospital Laboratory under the test name Complex 1 Assembly Western Blot Assay. The subcommittee recommends gap fill as no surrogate test was found. The closest potential crosswalk is 84181 protein Western blot with interpretation and report blood or other body fluid. Both align on methodology but vary on specimen type, which can impact resource usage.
Thank you. Do we have any other options, questions, or comments from panel members or CMS on this code? All right. If not, panel members, please cast your votes.
If you can just check your responses were logged, we're missing one. Perfect, thank you.
All right, next code up is this very similar code, 0655U.
This test is for inborn metabolic error of one enzyme using spectrophotometric kinetic assay on cultured skin fibroblasts. This test had no sponsor for the annual lab meeting and no presentation was received. After researching the assay, the subcommittee found that the test belongs to Children's Colorado Hospital Laboratory under the test name Complex 2 Enzyme Activity Assay. The subcommittee recommends gap fill as no surrogate test was found for crosswalk. The closest crosswalk is 84311 based on methodology alone. This specific test requires cells from a muscle biopsy, and due to the specimen type, additional pre-analytical resources are required prior to analysis.
Thank you, Jerry. Do we have any questions or comments from the panel or from CMS on this code? All right, if not, everyone please cast your votes.
Thank you.
All right, next up, 0656U.
Yes, this is a test for inborn metabolic error of one enzyme by radioactive activity assay on cultured skin fibroblasts. This test had no sponsor for the annual lab meeting and no presentation was received. And after researching the assay, the subcommittee found that the test belongs to Children's Colorado Hospital Laboratory under the test name pyruvate dehydrogenase enzyme activity, or PDH. The subcommittee recommends crosswalk to 82658. This is the strongest match since the two codes align on specimen type and methodology.
Thank you, Jerry. Does anyone have any comments from the panelists or CMS? Questions? All right. If none, panelists, please vote.
Thank you.
All right, and rounding out this set, we have formerly known as X266U with a final code of 0667U. And Jerry, this one's yours as well.
This test is for inborn metabolic error to determine fibroblast growth factor 21, or FGF21, concentration by ELISA using serum or plasma. This test had no sponsor for the annual lab meeting and no presentation was received. After researching the assay, the test donor appears to be Children's Colorado Hospital Laboratory. The subcommittee recommends crosswalk to 0251U hepcidin 25 by ELISA. Both tests are straightforward ELISA-based quantitative biomarker tests and are analogous to the FGF21 descriptor.
Thank you. All right, any questions or comments from our panel? Our CMS? All right, panelists, please cast your votes.
Thank you.
All right, we have some genomic sequencing procedures here. So next code up is 0647U, and Dr. Hooker Jillian will be presenting these.
0647U is an oncology MRD whole genome sequence analysis, cell-free DNA of whole blood. It's an assessment using a baseline patient-specific tumor profile, and the test is reported as a negative or % circulating tumor DNA. The sponsors recommended crosswalk to 0340U, which is also an MRD assessment of circulating tumor DNA. The panel also considered crosswalk to 0307U, also an MRD code, patient assessment follow-up to baseline. Notably, 0340U is an ADLT, and because of that, we recommend crosswalk to 0307U.
Thank you. Do we have any questions or comments from the panelists or CMS about this code? All right, if not, panelists, please cast your votes.
Thank you.
All right, staying with MRD and Jillian, we have code number 0642U up next.
Yep, 0642U is also an oncology MRD test, NGS-based in whole blood. Also a test to follow up compared to the baseline levels of circulating tumor DNA. We did not receive a recommendation for this one. It is a similar test, and we recommend crosswalk to 0307U.
Fantastic. Thank you. Do we have any other options, questions, or comments from panel members? From CMS? No? All right. If no additional comments, panel members, please cast your votes.
Thank you.
All right, next up, another MRD X294U with a new code of 0689U.
Yes, 0689U is also an MRD test, in this case specifically for colon cancer. It is a follow-up test using a patient-specific assay reported as a quantification of circulating tumor DNA presence and trends over time. The sponsors recommended crosswalk to 0340U, an MRD follow-up test. Similar to the above, the other crosswalk considered by the panel was 0307U for MRD. Subsequent assessment. Megan, did you say something? Sorry.
No.
Okay. Also, an assessment and follow-up to a previously analyzed sample. In this case, for the same reason as above, because 0340U is an ADLT and 0307U is not, the panel recommends crosswalk to 0307U.
Thanks, Dylan. Do we have any other suggestions, questions, or comments from panel members or CMS? All right, if not, panel members, please cast your votes.
Thank you.
All right, staying with MRD. Up next, we have 0645U. And I think I saw Dr. Jiang Jen come off mute, so this one must be yours.
Yeah, thank you. Yes, This is the code number 0645U for the oncology leukemia and the measure MRD detection. For the gene rearrangement, the methodology is digital PCR. The sponsor has done a great presentation, so we do agree with the recommendation for the gap fill for this particular code. Thank you.
Thank you. Do we have any other questions or comments from panel members or from CMS on this code? All right, if not, panel members, please vote.
Thank you.
All right, next up, we have 0641U, another MRD test.
Yes, I believe Mark gonna take this one. Thank you.
Thank you.
Yes, certainly. Presenting on behalf of myself and co-reviewer Dr. Jiang, this code was evaluated also without a dossier submitted to this panel. So 0641U is a MRD baseline test that uses NGS of a matched tumor tissue and blood to establish a variant reference for subsequent MRD monitoring assessments. And the panel recommendation here is a crosswalk to CPT 0306U. Similarly, both of these codes describe tumor-informed target NGS baseline MRD assessments using both the paired blood and tumor specimens to establish that reference.
All right, thank you. Do we have any comments, questions from the panel or CMS? All right, if not, panelists, please vote.
Thank you.
All right, I believe we're staying with Mark for the next code up, 0646U.
Yes, again, presenting on behalf of myself and co-reviewer Dr. Jiang, 0646U it's also an MRD baseline test, but in this case, it uses whole genome sequencing of a matched tumor tissue, white blood cells, and plasma cfDNA to establish a baseline for detecting circulating tumor DNA in stage 3 colon cancer. So the panel recommendation in this case is a gap fill, and that's consistent with the presenter's own recommendation. Unlike other baseline MRD codes reviewed in this cycle, 0646U uses whole genome sequencing across the 3 matched specimen types. And this methodology is much broader and more resource intensive.
Fantastic. Thank you. Do we have any other questions, comments about this code from either the panelists or from CMS? All right, if not, panelists, please vote.
Thank you.
All right, one more MRD test. We have X278U with the final code of 0688U, and Mark will be taking this one as well.
And once again, presenting on behalf of myself and co-reviewer Dr. Jiang, X278U, it's also a baseline test for colorectal cancer MRD that uses NGS of a circulating tumor DNA from whole blood and tumor tissue. And again, compare this against the subsequent MRD assessments. In this case, the panel recommendation is a crosswalk to CPT 0306U, which also describes a tumor-informed targeted NGS baseline MRD assessment, and that also uses paired blood and tumor specimens.
Fantastic. Any questions from panel or CMS? All right, if not, everyone please vote.
Thank you.
All right, and to round out this set, we have code number 0644U, and it looks like Jen will be taking this one.
Right, thank you, Megan. So for code number 0644U, this actually pairs with the previous code as 0645U because this current code 0644U is a baseline quantification test. It's using the personalized assay design as well as a methodology as droplet digital PCR. Therefore, we agree with the sponsor recommendations. This should be the gap fill code. Thank you.
Thank you so much. Do we have any questions, comments from other panelists or from CMS? All right, if not, panelists, please vote.
Thank you.
All right, and I believe rounding out our MRD codes, we have X262U with a final code number of 0693U, and I think Dr. Bridges is taking this one.
Yes, 0693U is also for head and neck oncologic tumors, and it measures circulating tumor DNA, and it uses a personalized panel, tumor-informed next-generation sequencing analysis for 699 genes of patient-specific somatic variants identified from primary tumor tissue, and the source is post-operative lymphatic exudate specimen, and the algorithm is reported as the presence or absence of circulating tumor DNA. There are 2 codes that this can crosswalk to. One is, so the matching codes are 0306U and 0560U, which are both tumor-informed MRD baseline codes with matching methodology. The 0306U is used for an initial baseline assessment to determine patient-specific panel for future comparisons, and the 0560U is allows for baseline assessment and construction of a personalized variant panel to evaluate current MRD and for comparison to subsequent MRD assessments. So we propose crosswalk to 06U crosswalk to 0560U.
Thank you. Do we have any questions or comments from the panelists or CMS on this code? All right, if not, panelists, please vote.
Thank you.
All right, next up, we have 0611U, and I believe Dr. Chung is taking these.
Thank you. Yes, it's me, my case. My code 0611U. This oncology for the liver with analysis of the over 1,000 methylation regions and use specimen of cell-free DNA. We do, there's no presentation for this case. Our subcommittee has did extensive discussion on this code, and we do believe crosswalk to 0565U is the most appropriate because it's similar methodology, similar specimen, as well as for the same organ disease. The only difference is that 0611U offers the, let me see, they offers the quantitative report, and 0565U report a qualitative report. So crosswalk is appropriate, and the gap fill is also should be within the consideration, but our panel does recommend crosswalk to 0565U. Thank you.
Thank you. Do we have any questions or comments from panel members or from CMS on this code? All right, if not, panel members, please vote.
Thank you.
All right, next up we have, I believe, the sister code to this code, 0612U.
Yes, thank you. Again, I'm going to repeat myself all over again. So.
All right, this one
Yes.
it's a identical
similar
clinical descriptor.
methodology, yes, and again, we recommend crosswalk to 0565U, and the gap fill is also could be the another alternative option, but our panel recommend crosswalk to 0565U. Thank you.
Thanks, Jen. All right, if we have any questions or comments from other panelists or CMS? If not, panelists, please vote.
Thank you.
All right, back to the Chem subcommittee. We have next up code formerly known as X292U, final code 0692U, and I believe Jerry will be presenting these.
Yes, this is a test for peripheral artery disease, analyzing 3 proteins by immunoassay using plasma, including an algorithm reported with the clinical results shown as a risk score for obstructive PAD. The subcommittee researched this test and recommends crosswalk to 0308U, heart, CAD, high sensitivity. Both tests share similar components, methodologies, and result type.
Thank you. Do we have any other questions, comments from panelists or from CMS on this code? All right, if not, please vote.
We're looking for 2 more votes. You could check that yours went through. There's 1 and 2. Perfect. Thank you.
Fantastic. Thank you. All right, next up we have 0640U, and Dr. Chung, Chris will be taking this one.
This is an oncology test looking for leptomeningeal metastasis using tumor cell selection. We agree with the presenting lab that it crosswalks well to 0338U. And that's the recommendation of the subcommittee as well.
Thank you. All right, does anybody have any questions, comments, from panelists or from CMS? All right, if not, everyone please vote.
Thank you.
All right, so next up, we have a series of similar services. I'm just going to read off all of the codes together, and then I think Dr. John can discuss them all together. So we have X261U, which is now known as 0681U, X263U, which is now 0680U, X265U, which is now 0684U, X273U, which is now 0682U. And that's what we have on the screen right now. So maybe if we do this,
We can do this 4 code at the same time. Then we
yeah,
can do
I think that's the best thing.
exactly. So everybody can see it. Yes. Okay. Thank you, Megan. So for all these 4 codes, as you can see, even though they are for various application like tobacco use or alcohol use disorder, as well as oncology, lung cancer, but the methodology or targets are the same. DNA analysis of methylation markers, use the digital PCR, and different specimens. However, we do believe because of digital PCR is a quite new technology, and we couldn't find any code appropriate in the current file to be crosswalk to. The sponsor did do a presentation and recommend crosswalk to 0566U, and we feel like 0566U is using the quantitative PCR, which is fundamentally different from the digital PCR. So we do not think this is appropriate code to crosswalk to. The panel recommend a gap fill for this, for all the actual 4 codes. Thank you.
All right, so I think we'll vote on each one of these in turn. Does anybody have any questions or comments from panelists or CMS on this set of 4? All right, if not, please vote on X261U.
Thank you.
All right, now X263U, please cast your votes.
Thank you.
X265U, please cast your votes.
Thank you.
And finally, X273U, panelists, please cast your votes.
Thank you.
All right, we have 2 more in this series, X286U, which is now 0683U, and X293U, which is now 0685U.
Thank you, Megan. Yes, those two codes, again, as you can see, they are using the methylation sensitive digital PCR, which is a very new technology. And again, the sponsor recommend crosswalk to 0566U after discussion within the panel members. And we do believe this should be recommended as a gap fill for both code. Thank you.
Thank you. Does anybody have any questions on these codes? All right, if not, panel members, please vote on X286U.
Thank you.
And now X293U, please cast your votes.
Thank you.
All right, next up, we have 0606U, and Dr. Chung will be presenting this one.
Right, we didn't receive a presentation or a presenter. The test is hematology red cell membrane test using active cytometry, and it's in our research we can crosswalk it to 0305U, which is also a red cell membrane disorder, and so that's the recommendation of the panel and subcommittee.
Thank you. All right, does anybody have any questions, comments from panelists or CMS? All right, if not, please cast your votes.
Thank you.
All right, next up, we have code number X259U, which has a final code number of 0665U. And Dr. Schmatzer Christine, I think this one's yours.
Yes, it is. Thank you. So X259U, now 0665U, is a multi-analyte test, a hepatology test for MASH, with one analyte measured by ELISA and another by RT-qPCR. The sponsor recommended crosswalk to 0468U minus individual CPT code fees for 83883 and 83036. Which are hemoglobin A1C and an individual nephelometry analyte. Those two analytes are not in this current recommended code. Thus, in our review, we do agree with the sponsor's recommendation and recommend a crosswalk to 0468U minus 83883 and 83036.
Thank you. All right, do we have any questions or comments from panelists or CMS on this code? All right, if not, panelists, please cast your votes.
Thank you.
All right, thank you. So the next grouping is sort of a set, but maybe we take it as they're presented on the screen here. That might be easiest for everyone. So we have 0616U up next, and Dr. Leonards will be taking these.
Yeah, thank you very much. Yeah, this is a set of over 10 codes. In this case, methylation profiling for the use case for all these is a different number of genes, all DNA methylation analysis, all from whole blood, all with an algorithm reported as positive or negative for risk. So this is not a score, that's somewhat important. Briefly, the subcommittee has discussed a total of 5 different potential crosswalks and ultimately recommends a crosswalk to 0565U, which is also next generation sequencing based. It's an epigenetic alteration assay from plasma with an algorithm reported as cancer signal detected or not detected. While the use case here for this whole set of assays differs, the risk, the algorithm output, and the overall complexity was the closest similar. The other codes did not match either the technique, the algorithmic output, or the overall effort as close to the subcommittee recommendation 0565U.
Thank you, Joe. So we'll vote on this one. Does anyone have any comments, questions from panelists or from CMS? All right, so let's vote on 0616U.
Thank you.
All right, next up is 0617U from the same submitter.
Correct. Same as presented before, with the difference that here a cardiovascular atherosclerotic risk is determined, also reported as positive or negative risk. Same recommendation by the subcommittee, crosswalk to 0565U.
Thanks, Joe. If anybody has any questions or comments from the panelists or CMS, please go ahead and vote.
Thank you.
All right, next up in the similar series, 0618U.
Correct. Again, same technology here. This use case is in psychiatry for positive or negative risk for bipolar disorder. Same recommendation by the subcommittee and myself to crosswalk 0565U.
Thank you. Unless we have any comments or questions from the panelists or CMS, panelists, please go ahead and vote.
Thank you.
Okay, we'll keep rolling with 0619U. Maybe we can just do both of these since they're on the screen together, Joe. So 0619U and 0620U.
Yes, same technology, differences in the total number of examined methylation analysis sites, but the two use cases, pulmonary chronic obstructive pulmonary disease and oncology hepatocellular carcinoma, which both match the crosswalk to 0565U. So first code here is 0619U for vote, and crosswalk as previously 0565U.
All right, thank you. Panel members, if you could vote on 0619U.
Thank you.
And now 0620U. Please cast your votes.
Thank you.
All right, and I believe we have one more code in this series. It is 0622U.
Yep, same assay, and I think there's a few more coming. Psychiatry, major depressive disorder reported as positive or negative risk. Same crosswalk to 0565U.
Right. Thank you, Joe. Panelists, please cast your votes for on 0622U.
Thank you.
All right, I lied. We are not finished with this series. We have 0623U and 0624U.
Mhm. Both again, same technology, same platform, both 5,000 sites methylation analysis. The first code now for autoimmune disease, in this case, multiple sclerosis. The second code that we're voting on soon is hepatology, NASH, non-alcoholic steatohepatitis. Again, for both the subcommittee and my recommendation is to crosswalk to 0565U.
Thank you. All right, panelists, could you please cast your votes on 0623U?
Thank you.
All right, and please cast your votes on 0624U.
We're just waiting on one. You could just check, please. Perfect, thank you.
All right, and then we can see, I don't sure, got one on this side. All right, so this is again in this same series of codes, 0625U.
In this case, the indication is endocrinology test for osteoporosis, again reported as positive or negative risk, crosswalk to 0565U.
All right, panel members, please cast your votes on 0625U.
Thank you.
All right, and next up, I believe is 0626U. Same,
Correct. Here,
same submitter.
that's correct. Same assay, 20,000 methylation sites, neurology indication for Parkinson disease reported as positive or negative risk. Crosswalk recommendation by the subcommittee and myself is 0565U.
All right, panel members, please cast your votes on code number 0626U.
Thank you.
All right, and I believe next up, this one is in that same series, I believe, 0621U. And I'm not sure, well, Dr. Bridges, are you taking this one?
I can, unless you would like to take that, Dr. Leonard's. I'm going to go with your same recommendation. This is code 0621U. It's True Diagnostics, TrueD methylation detection system for Lyme disease. It's a genome-wide DNA methylation assay run on whole blood. The laboratory profiles more than 10,000 methylation sites and applies an algorithm that reports as positive or negative risk result for Lyme disease. It is one of 12 near identical methylation classifiers that True Diagnostics has submitted. It's all built on the same array and the same analytical pipeline for pricing. My recommended ballot option is crosswalk to code 0565U, a next generation sequencing DNA methylation assay with 0020M, a genome-wide methylation array assay as an alternative. Each of these comparators is a DNA methylation analysis paired with an algorithmic interpretation. And so I would defer to Dr. Leonard's, who has recommended crosswalk to 0565U on the previous MDS-based test.
Fantastic. Thank you, Dr. Bridges. All right, panelists, does anybody have any questions or comments? If not, please vote on 0621U.
Thank you.
All right, and rounding up the series, 0627U.
Same platform, same submitter. The essay is in the field of psychiatry as a reported positive or negative risk for schizophrenia. Same crosswalk recommendation to 0565U.
Thank you. All right, panelists, please vote on 0627U.
Thank you.
Thank you.
All right, we're going to switch gears and go back to some microbiology and infectious disease. Our next code up is 0600U, and Dr. Baselski will be taking these.
Yes, finally I get to speak. 0600U is a highly multiplexed assay using real-time PCR to detect many different potential organisms and resistance genes, 95 total. There was no sponsor or recommendations from the public for this. So we looked for other highly multiplexed assays with complex microbial possibilities and came up with a crosswalk to 0593U times 2. This is keeping with historical precedent where we look at the number of reportable targets and the specimen type. So again, the recommendation is crosswalk to 0593U times 2.
Thank you. Do we have any questions or comments from the panelists or CMS about this code? All right, if not, panelists, please vote.
Thank you.
All right, next up, we have X264U with a final code number of 0666U. And Vicki, I believe this one's yours too.
It is X264U repurposed as 0666U is very similar to the previous discussion. However, there are only 55 targets, 27 microbial targets, and 28 antibiotic resistance targets, but using the same historical precedent, looking for the number of analytes and similarly complex specimen, we arrived at a suggested crosswalk to 0593U.
Thank you so much. Do we have panelists or CMS? Does anyone have any questions about this code? All right, if not, panelists, please cast your votes.
Thank you.
All right, and sticking with infectious disease, next up we have X287U with the final code number of 0669U.
This code X287U, now 0669U, is a droplet digital PCR assay for 3 distinct infectious agents that have similar clinical syndrome. There is a previous code that accounts for one of these agents, one such agent by that same technology, which is 0301U. Because this is 3 distinct agents, we recommend a crosswalk to 0301U times 3.
Thank you. Do we have any questions or comments from other panel members or from CMS? All right, if not, panel members, please cast your votes.
Thank you.
Awesome. Thank you, Vicki. All right, we're going back to genomic sequencing procedures. Our next code up is X255U with a final code number of 0672U, and Dr. Hooker will be taking these.
Yes, 0672U is a whole exome mitochondrial DNA sequence analysis for rare disease, including deletions, duplications, STR, and done on blood or saliva. This code is for the proband plus one comparator. And the panel, the sponsors recommended 0214U plus 1 unit of 0215U, which is the comparator previously priced. And the panel and myself agree with this recommendation for that crosswalk.
Thank you, Jillian. All right, does anybody on the panel or CMS have any questions about this code? All right, if not, panelists, please cast your votes.
Thank you.
All right, next up we have X303U with a final code number of 0673 or 4.
Oh yeah, there was a typo. It is 0673 in this case.
Thank you.
Yeah. So code number 0673 is exome plus mitochondrial DNA sequence analysis, deletions, duplications, SPR, blood or saliva, and interpretation of that test. In this case, it is the proband plus 2 comparator sequences. So the sponsor recommended 1 unit of 0214U, the proband sequence analysis, plus 2 units of 0215U, the proband comparators. And both the subcommittee and myself agreed with this recommendation for that crosswalk to those codes, 0214U plus 2 units of 0215U.
Fantastic. Do we have any questions or comments from panelists or CMS on this code? All right, if not, panelists, please cast your votes.
Yeah, we did.
Oh, we have some comments? Let's see.
Maybe not. Is there a question in the room?
We have a question?
No, there's no questions.
Okay.
Thank you.
Thank you.
We're waiting on one more vote.
Okay, I'm sorry. Yeah,
I think we might.
Okay, thank you.
Fantastic. Thank you. All right, next up in this sort of similar series, X250U with a final code number of 0674U.
Yes, in this case, it's 0674U. That's correct. This is a whole genome sequence, including mitochondrial DNA sequence analysis, small sequence changes, insertions, deletions, copy number variants, mobile element insertions, homozygosity, aneuploidy inversions, STRs, done on blood, saliva, or extracted genomic DNA, inclusive of interpretation and analysis. In this case, the test is for the proband plus 1 comparator. The sponsors recommended crosswalking to 0212U, which is the whole genome sequence proband code, and 1 unit of 0213U, which is the whole genome sequence comparator code previously priced. Both the subcommittee and myself agreed with this crosswalk to 1 unit of 0212U plus 1 unit of 0213U.
Fabulous. Thank you, Jillian. Does anybody have any questions from the panelists or from CMS on this code? All right. If not, panelists, please cast your votes.
Thank you.
All right, next up, we have X271U with a final code number of 0670U, another duo code.
Yes, in this case, it is a whole genome sequence analysis with a combination of short and long read sequence for single nucleotide variants, insertions, deletions, intronic variants, copy number variants, larger deletions, duplications, mobile element insertions, homozygosity, aneuploidy inversions, inclusive of mitochondrial DNA, sequence and deletions, STR repeats, methylation analysis, done on blood, saliva, amniocentesis, CVS samples, tissue, and inclusive of interpretation of the sequence. As you said, this is a duo proband plus 1 comparator. The sponsors recommended crosswalk to the whole genome sequence analysis, code 0212U, and 1 unit of the proband code. Oh, sorry, actually, something's not lining up with this one. Let me go back and see what was on the sheet. Sorry, I think the recommendation, we did not receive a recommendation for this one. I'm sorry. So the codes that we considered for this are not the codes I mentioned previously. They are 0035U codes, which is a whole genome sequence analysis with both the short and long read sequencing, as well as the proband whole genome sequence analysis 0336U. And we recommended 1 unit of each crosswalk to 0335U plus 0336U. Is that clear to
All right.
everyone?
So the ballot options are correct. The long
Yes.
descriptors for the recommendations
That's
are
correct. We should have included the long descriptors. If anybody wants to see them, I think they should be on the next slide, but
they are on the next with the next code. So just to be clear for everyone, we are voting on the ballot options, the codes that are listed there, crosswalk to 0335U and 336U.
yes, that's correct.
Awesome. Thank you. All right. Does anybody have any questions or comments from the panel or from CMS? All right. Panel members, please cast your votes. And if there are any questions about the long descriptor, as Jillian just said, they are actually in the next entry.
Okay, thank you.
All right, and as I said, next one up is X285U with a final code number of 0676U, and another duo code. And these are the descriptors for the codes that were just discussed in the previous code.
Yes, that's correct. 0676U is a whole genome sequence analysis, including small sequence changes, copy number variants, deletions, duplications, mobile element insertions, UPD, inversions, aneuploidy, includes mitochondrial with analysis of heteroplasmy, large deletions, STRs, and maternal cell contamination. So in this case, this is for fetal samples, and this is for the proband plus the maternal comparator. And in this case, we did receive a recommendation to crosswalk this to 0335U plus 0336U. The descriptions are there. Whole genome sequence, fetal sample, plus the comparator 0336U. There, and that was the submitted recommendation. The subcommittee and myself agree with that recommendation for the crosswalk to 0335U plus 0336U.
Does anybody have any questions or comments on the on this code? All right, panel members, please vote.
Thank you.
All right, so we've had some duos now. We'll talk about some trios. So we've got code number X272U with a final code number of 0675U.
Yes. So in this case, 0675U is a whole genome and mitochondrial DNA sequence analysis. In this case, it is a proband plus 2 comparators. The sponsors recommended crosswalk to 1 unit of 0212U, previously reviewed, which is a whole genome and mitochondrial DNA sequence analysis for the proband. In this case, because it's a trio, it would then be crosswalked to 1 unit of the proband code plus 2 units of the comparator code 0213U, which is the comparator for the genome and mitochondrial DNA analysis. So that was the submitted recommendation. The subcommittee and myself agreed with that recommendation and recommend that crosswalk.
Thank you. All right, does anyone have any questions or comments from other panel members or from CMS? If not, panel members, please cast your votes.
Thank you.
All right, and sticking with this grouping, we have X302U with a final code of 0671U.
So in this case, this is the genome sequence analysis trio in fetal samples. So analysis of blood, saliva, amniocentesis, CVS, and including interpretation in a fetal sample. The submitted recommendation for this is 0335U, which is the whole genome sequencing code for fetal samples, and plus 2 units of 0336U, which are the comparator codes for the genome sequence analysis in fetal samples. The subcommittee and myself reviewed this, agreed with that crosswalk recommendation of 1 unit of 0335U plus 2 units of 0336U.
If anybody has any questions or comments from the panel members or CMS? If not, panel members, please cast your votes.
Thank you.
All right, and next up, X291U with a new code number of 0677U.
Right, 0677U is whole genome sequence analysis, inclusive of sequence changes, copy number variants, del dupes, UPD, mitochondrial, heteroplasmy, large deletions, STR, and inclusive of analysis for maternal cell contamination. In this case, Megan, I'm wondering if 2 of them didn't get switched somewhere.
I was just looking at those, Jillian. I think we've got some prenatal, fetal, and
I feel like would it make sense for us in the break to go back and look at these and bring them up at the end of the meeting? Make sure
I was just going to suggest that. So
Okay.
just everyone, just to make sure that we've got the crosswalks and ballot options right. I think Jillian, I would like to suggest that we skip this one for now and come back to it and double check the other one as well to make sure that we've got the ballot options and the descriptors accurate.
Yep, agree.
All right. Well, that wraps up that one. We'll come back to those after the break at the end. All right, so next up we have 0657U And I think Heather will be taking these.
Yes, thank you. 0657U is again a rare disease focused rapid whole genome sequence analysis of the comparator nuclear and mitochondrial DNA by next generation sequencing, blood or buccal sample with the relevant variants reported along with proband results. So there was a recommendation by the submitter to crosswalk this code to 0425U. We considered multiple options as the panel, and we, as the panel, have recommended specifically a crosswalk to 0212U plus 0213U. The methodology, resources, etc., are virtually identical in these codes to the new code that's been obtained by this vendor. And so we, as a panel, recommend that crosswalk to 0212U plus 0213U. There are alternative options provided here, including abstain and gap fill.
All right, do panel members have any questions or CMS about this particular code? All right, if not, panel members, please cast your votes.
Thank you.
All right, next up, we have 0658U.
0658U, again, another rare disease whole genome sequence. The difference between this code and some of the other codes presented today that this is a rapid whole genome sequence analysis, but it does include all of the SNVs, insertions, deletions, CNVs, etc. in this test. The sponsor had recommended a crosswalk to 0094U, which is a previously priced genome sequencing, whole genome sequencing analysis with a rapid analysis. So that is one of the options for crosswalk. When the panel reviewed, we did identify alternative options, and the panel landed on a crosswalk recommendation of 0212U plus 0213U for this test. And then the other optional crosswalks, as well as abstain and gap fill, are available for selection.
All right, do we have any questions from the panel or CMS on these codes? All right, if not, panel members, please cast your votes.
Okay.
All right, next up, we have 0659U.
0659U is again in this series of whole genome sequence analysis codes for rare disease. The differentiator in this code is the ultra rapid approach to the test and the results. We had a recommendation from the sponsor of 81425 times 1.65. The 1.65 in their presentation accounting for the ultra rapid aspect of the analysis. So we have that option available for selection. As a panel, we've reviewed and recommended a crosswalk to 0212U plus 0213U for this code as well. And then, as always, we have the abstain, gap fill, and other potential codes for consideration as well.
Do we have any questions or comments? Any other options from the group? All right, if not, panelists, please cast your votes.
Okay.
All right, we're gonna switch gears and go back to some microbiology codes. We have up next 0607U. And let's see who's coming off mute to present this one.
I think I am. This is Vicky Zaselsky. This was a code evaluated by both subcommittees, but ended up, I think, in the realm of microbiology. 81 and 82 are very related codes. They are both codes for real-time PCR detection of many bacterial organisms that are part of the microbiome. They're done on endometrial biopsies, which is somewhat unique. And they're expressed as reported as quantified levels of bacterial organisms, not just qualitative. So for 0607U, 31 organisms, 31 bacterial targets are assessed. We looked at some other multiplex assays that looked at complex microbiomes like vaginal swabs, but these are quite different sample types than biopsies, and they do not give quantified results. So we felt that gap fill for 0607U was appropriate. And if I can throw in for 82, 10 organisms rather than 31 organisms, but again, by the same reasoning, there are no other codes on that specimen type that give quantified results. So that one would also be recommended for gap fill.
Thank you. Do we have any questions from panelists or CMS on these two codes? All right, if not, panelists, let's vote on 0607U.
Okay.
All right, and panelists, please now cast your votes on code 0608U.
Okay.
All right, back to oncology. Next up is code number 0562U, and Dr. Bridges will be taking this one.
And I have the 0562 0621U, correct? No, wait, 0562U, which is LabCorp's I think it's personal genome diagnostics, Elioplasma Focus DX, and FDA de novo authorized liquid biopsy test working from plasma cell-free DNA. It uses next-gen sequencing to detect single nucleotide variants and small insertions and deletions across 33 genes, copy number amplifications in 5 genes, and translocations in 3, down to a limit of detection near 1/10 of 1% variant allele fraction For pricing, my recommended ballot option is crosswalk to category 1 code 81462. The rationale is that this code describes a solid organ neoplasm genomic sequence analysis panel performed on cell-free nucleic acid from plasma, interrogating for sequence variants, copy number variants, and rearrangements, the same analyte, the same specimen, and the same methodology as 0562U, and therefore the closest match on resources. And I recommend the 81462 over I guess the company proposed 0388U because 0388U is a narrower non-small cell lung cancer plasma panel, and because CMS prefers a crosswalk to a category 1 code when it fits. But 0388U remains on the ballot as an alternative.
Thank you. All right, does anyone have any questions or comments from the panelists or CMS on this code? All right, if not, panelists, please cast your votes.
Okay.
All right, Alina, we're sticking with you. Next code up is 0648U.
So code 0648U is Thermo Fisher's Oncomine DX Express test, an FDA-approved comprehensive genomic profiling assay from fixed tumor tissue. It performs combined DNA and RNA next-gen sequencing across 46 genes, detecting sequence variants, copy number changes, and gene fusions on the rapid automated Genexus sequencer, and it carries an on-label companion diagnostic indication. For pricing, my recommended ballot option is to crosswalk to code 0022U. The rationale is that 0022U is a targeted DNA and RNA genomic sequencing panel for solid tumors that shares the same dual nucleic acid methodology and resource profile. I would flag for the panel that 0022U is a 23 gene non-small cell lung cancer panel, so it is a somewhat smaller comparator than this 46 gene pan-solid tumor assay. The panel may wish to weigh whether that difference in content warrants any adjustment.
Fantastic, thank you. Do we have any questions or comments from panel members or from CMS on this code? All right, if not, panel members, please cast your votes.
Okay, thank you.
All right, thank you, everyone. So I think at this time to give the folks enough time to double check some of our the ballots and the long descriptors that we have here. I know the agenda said that we were going to take a break at 12:30. We are going to start that lunch break now to make sure that everyone gets in a nice full lunch. And so we are going to pause at this time for lunch and then have everyone back. We will reconvene the afternoon session at 1:30 Eastern time. And I think I will turn over to Rashida if we have any additional comments for the panelists or attendees.
Thank you, Megan. Yes, we will break for lunch. And if you're a standby speaker or panelist, please reconnect by 1:20 with a 1:30 start time. Thank you all. Enjoy your lunch. Good afternoon, all. Welcome back to the CLT panel meeting. We're going to start in about 30 seconds. Before we begin, we just want to do a roll call of our panel members for record keeping. And just to acknowledge, this is Dr. Rashida Arthur with CMS, Designated Federal Officer for the CDLT panel. Okay, I'm going to read off panel members' names. Just please state you're present, just so we can have it for record keeping. Vicky Baselski,
Present.
Pranel Chandra,
Present.
Jerry Garner,
Present.
Chris Chung,
Present.
Jillian Hooker,
Present.
Megan Landsberg,
Present.
Jennifer Jang,
Present.
Jochen Lenners,
Hello, I'm here.
hi, Mark Rumpler,
Present.
Christine Schmacher,
Present.
Michelle Schoolmaker,
Present.
Heather Schappel,
Present.
Alina Bridges.
Present.
Thank you. Okay. Okay, I will hand it back over to Megan, where we'll be doing some continuation of our previous code discussion.
Thank you, Rashida. So just for everyone's knowledge, we did have there were a couple of errors in some of the codes that we have discussed. Some of the whole genome sequencing combination codes. We are making some corrections to those codes, and I think double checking to make sure that we are getting what the panel's actual recommendation was in both the ballots and the long descriptor. So I think for now, if it's okay with everybody, I think we are at number 85. And then if we can, what we'll do is just finish the rest of the codes, and then we can come back around to making sure that we've got those genome codes corrected at the very end, if that's okay with everybody.
Sounds good.
All right. So we are, I believe, this is where we ended with number 85. Am I correct? Sorry, everyone. It's only been an hour and it feels like yesterday. Okay, so I think we are at item number 85, code number X295U with the final code number of 0696U. And I believe Pranil Chandra was going to discuss this code.
That's correct. Thank you, Megan. So I had this code. It is an RNA sequencing-based code that entails an analysis of more than 300 genes to look at a variety of RNA-based alterations. There was a very nice presentation by the sponsor and a great outline and a rationale for crosswalk to CPT code 81456, which is, as you all know, it's the greater than 50 gene CPT code for RNA analysis. So it was very appropriate and apt and commensurate with the resources. And I agree with their recommendation, and that is our recommendation to crosswalk to 81456.
Fabulous. Thank you. Do we have any questions from any other members of the panel or from CMS? All right, panelists, please cast your votes on this code.
Shoot.
We are just waiting on one more vote.
Okay. I got logged out somehow.
Oh, okay. We'll wait. No problem.
What do I use as do I just put my email or my username? It wasn't taking my email.
Okay, so yeah, your name should just work.
It says error connecting with Google account.
Did you go back to my email from this morning with the link?
Oh, I already had the link, but then it logged me out. So, and then it asked for a password, which I don't know.
Huh.
Error connecting, so.
Have you tried to refresh your browser?
I did, yep.
You X out the window completely?
Okay, I just opened another browser. Sorry, I had to find the link. My name doesn't work.
And you're pointing your browser to the pollev.com?
Yep, I just went in and took the same link that you sent in previously. Now I'm just entering my email. Then it asks for a password, but I don't know that I have.
No, you shouldn't need a password. Everybody bear with us. We will figure this out.
Okay, I had to put in my Google account and that we're fine. Okay, sorry.
Perfect. All right, and there's your vote. Thank you so much. Okay, we're ready for the next.
All right. Now we're going back to infectious disease again. We are at 0601U. And I think, Michelle, were you taking these?
No, I'm taking this. This is Vicky Basowski.
Vicky, all right, all you.
These will be the last three micro ones you have to endure today. 0601U is a rather complex multi-analyte assay that looks at both biomarkers and microbial antigens using immunoassays as well as some synovial fluid, cellular analysis, and a diagnostic algorithm that's reported as a probability score. So it uses a whole lot of different methods. The sponsor and AdvaMed both agreed that a crosswalk to an oncology test, 0342U, was appropriate. But when we evaluated that code, although the biomarkers detected by immunoassays are somewhat similar, the reporting is quite different and the other components of the analysis are not there. So we felt that a gap fill, because of the complexity of the analyses done in this prosthetic joint infection assay, it was indicated. So our recommendation is gap fill.
Thank you. Do we have any other panel members that have any questions or comments on this code? Or any questions from CMS? All right, if not, panel members, please cast your votes.
Okay, thank you.
All right, next up is 0610U.
0610U is an assay that is performed on positive blood culture. So it's an add-on to a blood culture that looks at phenotypic antimicrobial susceptibility testing on those positive blood cultures. It uses a microfluidic sensor technology, so is different than the only other phenotypic positive blood culture code that is in the CLFS, and that would be the one that is for the Accelerate assay. So we did not feel, because of the different methodology, we did not feel that that other phenotypic blood culture assay was an appropriate crosswalk, and we recommend gap fill.
All right, thank you. Do we have any panel members or CMS who have any questions or comments about this code? All right, if not, panel members, please vote on 0629U. Oh, sorry. 0610U is the code that we are voting on right now. Apologies.
Okay, thank you.
All right, now we are on 0629U.
And this is the final microbiology code for the day. It is infectious disease detection of Mycobacterium tuberculosis using a CRISPR technology. This is a technology that's not reflected in any other infectious disease analytical tests that are currently in the CPT book. So we felt like this was clearly a gap fill recommendation.
Fantastic. Thank you, Vicki. Panel members, does anyone have any questions from the panel or from CMS about this code? All right. With that, panel members, please cast your votes on this code.
Okay.
All right, next up we have X269U. Final code is now 0694U. And I believe Dr. Chung is going to tell us about this code.
Right. So this is a oncology monoclonal gammopathy test using multi-TOF mass spectrometry and determining the isotypes and the M protein concentrations. This is a semi-quantitative test. The submitter is correct that 0077U is not an equivalent because this is a qualitative, that one is a qualitative. So the panel and subcommittee agree that the recommendation is gap fill.
Thank you. All right, does anyone on the panel or from CMS have any questions about this code? Right. If not, panel members, please cast your votes.
Thank you.
All right, so we're switching back over to some genomic sequencing procedures, starting with code number 0628U. And Heather's going to be discussing these.
Sorry, just getting myself off mute there. 0628U is a genomic analysis for a rare renal disease panel using saliva. It's a next generation sequencing assay of many genes with the report indicating pathogenic, likely pathogenic, or variants of uncertain significance. The sponsor recommended a crosswalk to 81455, which is actually a solid organ neoplasm test of 51 or greater genes. And the panel reviewed this code and reviewed the other CPT codes that were crosswalk options. And it is the panel's recommendation that the most representative crosswalk in terms of resources, methodology, et cetera, to this 0628U assay is actually CPT code 81432. So the panel's recommendation is to 81432. The sponsor recommended 81455, and then of course we have the alternatives, abstain or gap fill.
Thank you, Heather. Does the panel have any questions or comments about this code, or does CMS have any questions or comments about this code? All right, if not, panel members, please cast your votes.
Okay, thank you.
All right, next up we have 0651U.
Yes, this is Heather again. This is my code assigned to the molecular genetics subcommittee. This is an oncology genomic DNA test looking at hereditary cancer predisposition genes using next generation sequencing and MLPA to identify variants of clinical significance. There was no recommendation, I believe, on this one from the sponsor or from any professional societies. And after reviewing this code and looking at the options available to us, our recommendation for crosswalk is to 81432.
Thank you. All right, do any panel members or CMS have any questions or comments about this code? All right, if not, panel members, please cast your votes.
Thank you.
All right, next up we have another hereditary panel, 0653U.
Oh, yes. Sorry. This is again me. This is another nephrology. Oh, wait.
Yep.
0553U. Yeah, this is another nephrology next generation sequencing panel of a large number of genes, identifying pathogenic variants or likely pathogenic variants using whole blood, saliva, or they also say nail clipping. There was no recommendation from the sponsor or from any of the professional societies in terms of crosswalk for this code. The panel reviewed the available options or multiple options and recommends a crosswalk to 81432 for this code.
Do we have any questions or comments from the panel or CMS on this code? All right, panel members, please vote.
Okay.
All right, next up is a category 1 hereditary code, 8XX19.
Yes. This code represents a next generation sequencing test that includes sequencing and copy number variant analysis for 3 genes associated with Alport syndrome. We, as a panel, looked at available options and also considered the crosswalk that was recommended by the 2 professional societies that submitted recommendations. Those professional societies recommended a crosswalk to, and I'm not sure I'm going to say this right, 81407 plus 81408 times 2, and then the whole thing times 0.5. We looked at that recommendation. We looked at the tier 2 codes that are referenced in that recommendation, and we searched for a representative tier 1 code and identified 81432 as the most comparable assay. And so the committee recommends, or the panel recommends 81432 crosswalk to this 8XX19 tier 1 code.
Okay, do we have any questions from the panel or from CMS about this code?
I had this is Michelle. I had a question about just a matter of process here. Are we allowed to use the tier 2 molecular codes, or are these instead, I think we've historically treated them as the unlisted or not codes, because of the large, buckets of tests that fall under them?
Yeah, I think they were included here because they were a public recommendation from stakeholders.
Yes.
Yeah, that's why they're here.
Yeah, it's definitely my understanding that whatever a stakeholder recommends has to be one of the options in our pick list. And that's why it's there.
Okay, because I noticed we didn't put, I think it was 0629U, we didn't add their recommendation for 0077U. We just had the gap fill and abstain.
Oh.
Which number is that one?
I think it was, take a look at 0629.
Yeah, I can answer that. Michelle, they didn't actually they said they mentioned 0077U, but said it wasn't appropriate, and they wanted a gap fill.
Okay. So that wasn't actually, that was considered by the subcommittee, but not actually put forward by the sponsor.
No, the
Okay.
sponsor didn't even want what they mentioned it. They didn't think it was appropriate.
Okay.
Awesome. Thank you, everybody. All right, do we have any other questions about this code? All right, well, panel members, please vote on this code.
Okay, thank you.
All right, next up, we're sticking with hereditary panels. We have X254U, which has a new code of 0679U.
Yes. So this code is again a hereditary cancer panel with it's stated here whole genome sequencing in this assay, including SNVs, deletions, insertions, intronic variants, copy number variants, etc. The sponsor of this code had recommended, I believe, I have this right, 0214U plus 0215U as one option to consider. There was also a recommendation from the sponsor that I saw of 0218U plus 81435U, which I'm actually not seeing here as an option. But I believe that that was another option for crosswalk recommended by the sponsor of this code. So if we could check on that and just make sure that shouldn't be here as an option. The panel reviewed these recommendations, reviewed potential alternative codes that may be similar, and we as the panel are recommending a crosswalk again to 81432 for this code, or consideration of gap fill based on the whole genome sequence statement in this code.
Dr. Chappelle, what was the crosswalk suggestion that you said was missing?
The one that I saw, I thought in this one was 0218U plus 81435.
I think this submitted recommendation was 214 and 215, right?
Oh, I saw
I think
I
that's
saw-
the, I
That's
think that might be,
correct.
that's the next code. that we're-
Oh, sorry, then I have them maybe written in my notebook too closely together, and that could be the problem. I have like 3 different things up here, so I might have just read it off on the wrong
No
one.
worries. Yeah, I think that's the next code. You
Okay,
know, so
got it.
just, yeah, so for the panel, there's a number of different options here, right? So we've got gap fill, a couple of crosswalks, and then either crosswalking to other PLA codes or crosswalking to the Cat 1 hereditary panel 81432. Does anybody else have any questions? Panel members, CMS? Looks like we
We have some Q and A's and a hand up on this one that the choices are not what was recommended.
Okay, so we might be, so we are missing what the crosswalks are from the stakeholders. Okay, so for this code, all right, sorry, I think we have the. So Shruck, we
Hello.
she's a, yeah, can we turn
Can
her?
you hear me?
Yeah, there we go. Thank you.
Hello. Thank you. I have with me some of my colleagues from the sponsor, from the stakeholders' perspective, and this is not what was recommended for this test. For this text, we actually recommended 0218U plus 88233 plus 81441 for the skin biopsy part. And none of this, as I can see here, is on the screen.
Correct. Okay.
Yeah, I mean, in the spreadsheets we're working from, I didn't see that crosswalk in any of the codes that were assigned to me. And it still looks, if I go to this line in the spreadsheet, that we had it recorded as 214U plus 215U.
So 214U and 215U are for whole genome analysis, and that started with rare disease, hereditary, etc. So, and on the, like, I will double check the spreadsheet. I'm sorry if this is a mess over there, but on the presentation that we did a month ago, we for sure presented that.
Ms. Wise, could you state your recommendation?
Yes. So our recommendation are 08218U plus 88233 plus 81441. 3 different codes. This plus that.
Could you repeat it again?
0218U 88233 and 81441.
Okay, thank you. Dr. Landsberg, what I think we should do is put this code toward the end, and we could do a revote at the very end, or vote at the end.
Yes, I agree. And Cheryl, could you give us a rationale for having those particular codes? So 0218U, is my understanding, is the full gene sequencing for DMD on top of a different disorder. Was there a particular rationale for why that code should be separate from the others?
Yes. Can I add some of my colleagues to be here as well, to be the presenter of the codes with Dr. Christine Stanley? And let me also grab this and I'll
Sure. How about we'll give you a couple of minutes to get a hold of Christine, and then we'll keep going on, and then we can come back to this one when we have everybody on, just to keep the meeting moving, and then we'll come back and we'll readdress this code.
Okay, perfect. Thank you so much.
Gotcha. Okay. We are at X28U. U, which is now 0678U. And I imagine this one is going to be very similar, right? So this is from the same submitter. Cheryl, could you, I believe this might be a similar situation as the last one? Genome sequencing for a hereditary cancer panel? I think Cheryl might have disappeared on us.
I think Cheryl's here. Jolene Dalton.
Oh, sorry, I was on mute. I'm so sorry about that. So I said yes, thank you for bringing this up again. Also, some of our previous codes were discussed to be at the end of the meeting because some of the crosswalks were wrong. So let me make sure that my colleagues are here, Christine and Jolene, and yes.
We're here. We could just take this at the end with all the other codes if you wanted to.
Okay.
Let's do that because I think the other codes we're going to revote on those for sure.
Okay. So
and then we can take a look at these two when we're doing those other two will be relatively quick.
Perfect. Thank you so much. Thank you.
Okay. Thank you all for your patience. Okay, so we'll come back to these two, 0679U and 0678U. We'll move on, to keep the meeting going here. Our next code is a tier 1 code, 8XX17. And I believe Dr. Bridges is going to discuss this one.
So code 8XX17 is a genomic sequence analysis panel for heritable immune disorders. It sequences at least 15 genes and includes copy number analysis, and it is a non-proprietary category 1 panel rather than a single laboratory's proprietary test. For pricing, we recommended ballot option is a crosswalk to category 1 code 81432. The rationale is one of methodology and resources. 81432 is a defined multi-gene germline sequencing panel that likewise includes both sequence variant and copy number analysis. So the laboratory work and reagents are comparable regardless of which disease genes are interrogated. The alternative proposal by the American Clinical Laboratory Association is 81443, which is also a multi-gene sequencing panel and remains on the ballot. I favored 81432 as the closer resource match.
Thank you. Do we have any questions from our panelists or CMS about this code? All right, so panelists, please cast your votes.
Thank you.
All right, we have another tier 1 code, 8XX22, and I think Alina, this one is yours as well.
Yes, it's code 8XX22. It's a genomic sequence analysis panel for complement-mediated thrombotic microangiopathy, atypical hemolytic uremic syndrome, and G3 glomerulopathy. It sequences at least 8 genes and importantly requires analysis of the structurally complex CFH and CFHR genes, including their rearrangements. It too is a non-proprietary category 1 panel. For pricing, we recommended the recommended ballot option is a crosswalk to category 1 code 81432. For the same reason as the previous panel, 81432 is a multi-gene germline sequencing panel with combined sequence variant and copy number analysis, which matches the methodology and resource use of 8XX22, including the added burden of structural analysis at the complement locus. The American Clinical Laboratory Association alternate is 81412, and it remains on the ballot. I recommend 81432 as a closer match on complexity and resources.
Thank you. Do we have any questions from the panelists on this code or from CMS? All right, thank you, everybody. Panelists, please cast your votes.
Thank you.
All right, next up, we have X279U with a new code number of 0691U. And Dr. Hooker will be taking this one.
Hi, yes. 0629 or 0691U, carrier screening for cystic fibrosis, SMA, the hemoglobinopathies, alpha thal, and DMD. So 6 gene totals. The sponsors recommended crosswalk to 0449U, which is carrier screening for inherited conditions. In that case, 5 genes, but in general, the overall resources for the two should be similar. And so the subcommittee discussed and agreed with the sponsor's recommendation of crosswalk to 0449U.
Fantastic. Panel members or CMS, do we have any questions on this code? All right. Thank you, everyone. And panel members, please vote now.
Thank you.
All right. Next up, we have X253U with a new code of 0690U. And I believe, Heather, this one is yours.
Yes. So this code X253U has now the official code 0690U. We had a recommendation for a crosswalk on this code from one of the professional societies with a crosswalk to 0493U. And the committee reviewed that recommendation, also looked at other potential crosswalk codes and identified 0055U as a potential crosswalk code as well. And let me go to another page. The recommendation, hold on. The recommendation from the committee aligns with the recommendation from the professional society. The crosswalk to 0493U is recommended. And alternatives are provided for selection.
Thank you. All right. Do we have any questions or comments from other panel members or CMS about these codes? All right. If not, panel members, please cast your votes.
Thank you.
All right, next up we have 0643U, a cell-free circulating tumor, and I think Heather has this one as well.
Yes. So this is, this is an oncology cell-free DNA test looking at over 200 genes through next generation sequencing, looking at SNVs and insertions, deletions, copy number, as well as tumor mutation burden. This is using a urine sample, which is different than the existing codes that are on the crosswalk. But in terms of resources and methodology, there was a recommendation by the sponsor to crosswalk to, hold on again, I'm sorry, I just have things going on too many pages. I believe it was the sponsor's recommendation or the public recommendation to crosswalk this code to 0539U. As the panel looked at 0539U, we identified an alternative potential crosswalk of 81464. It looks like historically 81464 was the crosswalk code for 0539U, so that all aligns. The panel is recommending the crosswalk to the tier 1 code 81464, and the alternative options are available.
Thank you. All right, do we have any questions from the panel? Oh, okay, we've got, I think we have a question. Do we have a question from the panel?
Hello, this is Zoe from the sponsor. I think our original crosswalk recommendation is crosswalk to 0539U times 1.3 because actual resources is needed for extraction and also sequencing.
I see. So you are requesting an additional modifier for extracting DNA from urine as opposed to blood?
Yes.
Okay. What we can do, if we can put that on the ballot, right? We can add that if you would like. So, if we add an option 5 for the panelists to choose from, are we allowed to do that, team CMS? A 0539U times 1.3 from the submitter.
Say it a little slower.
So Zoe, you had 0539U times a 1.3x modifier.
Yes.
Is that correct?
Yes.
Okay, you should see that as option E now.
Oh, we're not seeing it
Just
on the screen.
Just for clarification, it won't be on the screen. But it is on the ballot, and when you see results reported out from this, you will see it on the ballot. We just don't have the capability to change the chart that's online on the fly.
Could we repeat that one more time? Which code is it? And then the ballot
This
shift.
is 0643U, and we've added a 5th option to crosswalk to 0539U times 1.3.
Yes, and this is Heather just confirming it's there on the ballot as an option for the panel members to select, in addition to the other codes I mentioned.
That is correct.
Just to repeat, the 5th option is crosswalk to 0539U times
1.3.
1.3. All right, so to clarify for all the panel members, we have our always there, number 1, abstain, number 2, gap fill, 3 is crosswalk to 0539U, 4 is a crosswalk to 81464, and 5 is a crosswalk to 0539U with a 1.3x modifier. All right. Thank you very much, everyone. So panel members, please cast your votes.
And just to confirm, you should see that ballot option visible on the screen.
We
And
do.
all the votes have been recorded. Thank you.
Thank you, everyone. All right, next up, we have 0602U, and I believe this is Dr. Leonard's.
Thank you. This is an endocrinology combined or mixed code with a multiple analyte algorithmic analysis composed of a digital droplet PCR methylation assay, a C-peptide immunoassay, and then a serum-based HbA1c immunoassay, all from whole blood, reported as an algorithm risk for diabetes mellitus. The panel discussed what that would look like. Unfortunately, there is no single gene methylation code by digital droplet PCR available. The closest we found is an MGMT assay, and then we would still need to stack 2 additional category 1 codes. And after review of this, the subcommittee as well as myself felt that gap fill is the best representation of this combination of tests. So recommendation is gap fill.
Thank you. All right, so does anybody have any other questions, panel members or CMS about this code? Not too much to select from here. So panel members, please cast your votes. All right, the next one up is 0649U, and this one goes to Joe as well.
This is a neurology test for Alzheimer's disease. It's a targeted next-gen sequencing assay based on DNA for 81 and 82 target regions from whole blood and a prognostic algorithmic analysis reported as a categorization of the so-called cognitive status. We reviewed this. There was a suggestion that a similar Barrett's esophagus risk score looked similar in terms of effort and genes. So that's a crosswalk option, which is 0506U. However, the recommendation from the subcommittee and myself is to go for gap fill because it's not perfectly represented, and this is a distinct disease, and it's not tissue-based, it's whole blood-based. So final recommendation from the subcommittee and myself is gap fill.
Thank you, Joe. Does anyone have any further questions on this PLA code from the panel or the CMS? All right, if not, panel members, please cast your votes.
Thank you.
All right, the next one up is X268U with the final code of 0686U, and this one also goes to Dr. Leonard's.
0686U is an oncology test in the field of prostate cancer, mRNA-based next-gen sequencing, expression profiling of 22 genes. Sounds at first glance, because it's reporting a risk score relatively similar to Oncotype DX upon review of the entire clinical lab fee schedule. The best match by effort and reporting is a crosswalk to 81542, which is also an oncology prostate-based mRNA microarray-based gene expression profiling assay with 22 genes. And that is the final recommendation by the subcommittee as well as myself, 81542 as a crosswalk.
Thank you. Are there any questions or comments on this code? All right, if not, panelists, please cast your votes.
Thank you.
All right, next up we have X298U, which has a new code of 0697U.
That is an obesity test of a DNA genotyping analysis of up to 41 genes from buccal swab or blood specimen. There's also some patient biometrics flowing into a risk score algorithm to identify a predisposition to up to 4 phenotypes, and it is a likelihood to benefit from therapeutics, which is a very interesting and novel approach. So we reviewed several crosswalk options. There are several components here that I believe warrant a little bit deeper dive. So 0349U is one of the potential crosswalk options. This is a drug metabolism, or for multiple conditions, also from whole blood or buccal. So that matches. Then there's 27 genes reported. There's variant allele analysis, and then a phenotype that may impact gene-drug interactions. So this part is fairly similar to the response or benefit from therapeutics. And then the other recommendation is 0466U. And 0466U is a genome-wide association type code that represents many of the traits better. So we had several discussions. The final crosswalk recommended by the subcommittee is 0466U.
Okay, do we have any questions from the panelists or from CMS about this code? All right, thank you, everybody. Panelists, please cast your votes.
Thank you.
Thanks a lot.
Thank you, Joe. Next up is 0603U, and I believe Mark has this one.
Yes, thank you. For this code, notably that there's no dossier submitted to the panel by the sponsor. But 0603U is a urine-based presumptive toxicology screen. It covers 77 drugs or metabolites. It's performed by liquid chromatography and tandem mass spectrometry, and the results are reported qualitatively. The panel recommendation here is a crosswalk to CPT 80307, which shares an identical methodology, indication, specimen type, and produces the same qualitative result as 0603U.
All right, does anybody have any questions on this code from the panel or from CMS? All right, if not, panelists, please cast your votes.
Thank you.
All right. Next code up is 0613U. And Mark, we will continue on with you.
Yes, 0613U is a urine-based test for DNA methylation and mutation status. It occurs across 6 biomarkers to generate a probability index for bladder cancer and urothelial carcinoma. The panel recommendation is a crosswalk to CPT 0420U because both tests serve the same clinical population with overlapping biomarkers. One thing I would note for the panel is that there's pending MAC coverage. There's a policy that's reviewing this clinical category that's would say confirm that every peer comparator platform uses digital PCR or real-time PCR rather than a targeted NGS, which is used by 0613U. So this makes it a methodological outlier, but nevertheless, the recommendation is still a crosswalk to CPT 0420U.
Thank you. Do we have any questions or comments from the panelists or CMS on this code? All right, if not, panelists, please cast your votes.
Thank you.
All right, next up we have 0632U, and Dr. Chandra, I think Punel is taking this one.
Thank you, Megan. So yes, this is 0632U. This is a next generation sequencing code for sequencing of red blood cell antigens, and it's through cell-free DNA, and it's on plasma. There was a nice presentation by the sponsor and very good rationale. And so we do agree with their crosswalk recommendation of 0488U.
Thank you. Do we have any questions or comments from the panel or from CMS on this code? Right, if not, thank you, everyone. Panelists, please cast your votes.
Thank you.
All right, next up, 0633U, and Pernell, this one is you as well.
Thanks, Megan. Again, this is 0633U. This is an OBGYN cell-free DNA assay looking at a number of targets. And it is another assay that and CPT code that was accompanied by a very good public presentation with very good rationale, which we happen to agree with. And so our recommendation is going to be to crosswalk to their recommendation, which is 0489U.
Fantastic, thank you. All right, do we have any questions or comments from the panelists or CMS? If not, panelists, please cast your votes.
We are just waiting on one vote. Oh, there it is. Thank you very much.
All right, perfect. Next up, X256U with a new code number of 0663. Dr. Chandra, keep it going.
All righty. So this is a CPT code that describes an obstetrics NIPT-based cell-free DNA procedure that's basically detecting the presence or absence of antigens that are sequenced and reported as detected or not detected per antigen. In contrast to some of the other CPT codes that I evaluated, there was no presentation, no dossier, but thankfully there was another CPT code that was very similar in resources that we were able to identify, and therefore we recommend a crosswalk to 0488U, which is commensurate with the resources required to deliver the assay result.
Great, thank you very much. Panel members, anybody have any additional questions, comments, CMS? No, if not, all right, panel members, please cast your votes.
Thank you.
All right, everyone, this is the last one, and then we'll go back and we'll revote on some of the genomic codes. So last code is 0631U. And Dr. Chung, Chris, this one's yours.
Thank you. So even though this is titled as oncology solid tumor, this is actually a test to identify clonal hematopoiesis. And so the sponsor's proposed crosswalk of 81162 is not appropriate. So the subcommittee and panel is recommending gap fill.
Thank you. Does anybody have any questions or comments? All right, if not, panel members, please cast your votes on this code.
I'm sorry, this is Heather. I do think that in the sponsor's presentation to the committee, there were additional codes that were recommended in the crosswalk.
I think you are correct. That's a good point. And I believe you are correct, Heather, that there should be additional codes from the public recommendations. If we could, well, let's check on those.
Yeah, if you look in the comment tracker, there's 81443 mentioned. The 81162 that's on the ballot, but 81443 is not on there, as well as 81462.
Let's see.
Dr. Leonard, could
Those-
you please repeat those two codes?
So the cell that I'm looking at is Q112, and the codes are 81162, which is on the ballot, 81443, which is not on the ballot, and then the last code that was presented is 81462. But as Chris said, these codes are different from the scope and the description of the assay used. But I think for transparency, I think we should include these as options in the ballot. I concur with that.
Yeah, they're now reflected on the ballot that the panel is seeing. Again, not on the chart that's on the Zoom slide, but it is on your ballot. And then we will update the rest of the materials after the meeting.
Okay, thank you, Heather. So we've got the gap fill, we have the crosswalk to 81162. There is a crosswalk to 81443. And the other one, was it a 0631U? Did I get that right?
It was 81462, I believe.
That's correct.
Okay, thank you.
We are waiting for 2 more votes, but we will take time so people can look at those codes.
Yeah, this is Michelle. Joe, did that change your recommendation at all?
No, Michelle,
I
actually,
think
that's
it's Chris's.
Yeah, it's my
sorry, Chris.
Yeah, those are all solid tumor codes, if I recall correctly. And so, no, this is to identify clonal hematopoiesis, which is a hematopoietic disorder.
Okay.
I mean, the just to support what Chris said, the 81443 is a specific 15 gene inherited condition code, which does not fit clonal hematopoiesis. I mean, in the furthest sense, that has something to do with genes, yes, but that's different. And then the 81162, it entails genotyping of genomic DNA for BRCA1 and 2, which they mentioned that it's in their clonal hematopoiesis assay, but that assay 81162 is completely different. It has a dual component with deep sequencing, etc. So I concur with what we discussed in the subcommittee and with Chris's recommendation that this is not reflected by anything else. Yep.
Am I correct in understanding that this is a combination of cell-free DNA, like plasma-derived cell-free DNA in the test? It's not actually looking at the tumor tissue, right? And then also looking at leukocyte DNA, is that right?
So as I think Chris mentioned it is classified as an oncology solid tumor, but in reality it is detecting chip. like clonal hematopoiesis of indeterminate. Yeah.
And it's doing that because it's looking at cell-free DNA and
I don't think they mentioned that it's
leukocyte-derived genomic DNA. It's a combination of cell-free DNA and leukocyte-derived genomic DNA.
they say blood reported as tumor-derived or non-tumor derived. I do not know what that specifically means, but to me, this is chip assessment.
Right.
Yes,
It's a chip assay. It was on
it's sort
Buffy
of like
Pope.
it is a tumor normal profile. However, it is targeting the chip component within the quote unquote normal blood sample. So when you're doing the comparison, but it's specifically targeted to identifying chip alterations, which would be at a lower variant percentage than say known germline variants.
Yeah, got it.
Sorry about that, everyone.
Yes, I feel like I want to count to 3 because the votes people have been rethinking, but they're stable now. So I think we're good.
Let's just reconfirm the options. They should be visible on the screen for code 0631U.
Yep, those are what we have on the ballot. Yes.
Okay.
How are we doing, everyone?
Okay, so we're doing great. We have finished at least a first pass. We do have some that we want to go back to. So Dr. Landsberg, I'm gonna look to you for the specifics. We have in our notes essentially revoting all the way from 70 to 80. Is that?
I think we can start at 73. I think if we start at 73, and I think we've got the folks from Variantics on the phone. I think one of this we do have a question for the group. Thank you, Cheryl. So I think where one of the confusions was where we have different crosswalk and descriptors. Let's see if we can go. Oh, too far. Go back to 73. Yeah. So we did have a question about the difference between 0670U and 0676U, which is the code right after this one. The long descriptor of this particular code includes amniocentesis and CVS as part of the specimen types within the code descriptor, which sort of implies prenatal testing. Because that's what your test. So was the idea to submit this one prenatal as well, even though 0676U has sort of been defined as fetal testing. This one is not. So the panel was trying to get a bit of clarity on the intended use patient populations that these two different codes were utilized for. Is one going to be strictly for fetal prenatal testing and the other not?
Yes, so thank you so much for the opportunity to talk and to explain that. So we have two different platforms. One is a whole genome short reads analysis, and for that we have a specific codes that we ask for, and another platform that is actually short and long reads whole genome analysis. So it's two sequencing methodologies. One for one is short reads and one is long reads, and combine them together, we allow better detection of the genome and getting more information. So for this specific, for 73, which is represented right now, this is for rare disease, whole genome sequence analysis combination of short and long reads. And this is actually for blood, for saliva, amniocentesis, and CVS. So this is not just for prenatal sample. This is for everything. Basically the combination of short and long reads. The other test that you were discussed that is only for prenatal is the X285U and the X291U, which is number 99 and 102, and those are prenatally only, and those are whole genome short reads only.
So I think those had the right crosswalks.
Okay.
So this one, previously the ballot options were a crosswalk to a combination of fetal prenatal codes. These are the correct codes now. So this one that we're looking at, we have crosswalk to 0212U plus 0213U. One is a proband, the other is a comparator. So we're essentially talking a regular duo service.
So this one, so for this one, we did not submit a recommendation because we wanted to submit a gap fill to represent the two sequencing methodologies. And the combination of 212U and 213U is actually more relevant for the X250U, which is whole genome short reads analysis for proband and comparator. So and the gap fill that we want to submit for this one is actually 0212U twice, because we're doing dual sequencing, plus 0213U for a comparator. So we would like to ask for the committee to add here another option for crosswalk for twice the 0212U plus 0213U.
Okay. And there's another code for short and long read, right? That we reviewed last year. Which code is that?
That was the X302U, and that was for basically for the short and long analysis for proband and two comparators.
That was this year.
Yes.
Yeah. Okay.
Yes, we have a lot of codes.
And the recommendation for a 0212U times 2 is because you were sequencing the proband twice?
Yes, exactly.
Once by short read and once by long read. But
exactly.
There's a code that got priced last year, and I just can't remember which code it was. That was short read and long read. Is that correct?
Yes. So
which
this
code
one
is that?
0567U. That was priced last year, and we would like to we wanted to submit like reconsideration for this one and gap fill and explain that. But that will wait for the next meeting, I assume.
Okay. So this isn't a reconsideration of that code. This is a different code.
Yeah, this is a different because this is a proband and a comparator.
Okay. But the proband code is one that was priced last year, but you're going to submit for
Exactly.
reconsideration?
Exactly. Yep.
Okay.
Okay. So for the panel, we have the submitter. So what I'm hearing, Sherelle, is that you are recommending, your recommendation is gap fill, but we also have options from the panel of a crosswalk to a proband plus a comparator, but you're also recommending a proband ×2 plus a comparator for a duo.
Exactly.
Okay.
And then just to clarify one more methodological thing. So it's two sequences, one long read, one short read, one analysis. They're not separately interpreted.
Yes, one report that includes short reads and long reads with all the added value of both sequencing methodologies. And there are two different codes here, one with one comparator for duos and one with two comparators for trio.
Okay. Thank you.
Thank you. And just for our panel members, for sake of understanding, is there, you're doing two different methodologies, to get to one answer. Is there, how do I ask this? What is the added benefit of doing the additional sequencing, of doing it twice, essentially? So there's clearly, some things that the long reads pick up and the short reads pick up. How is it additive, the information that you get?
Yes, yes. So I can explain a little more. So with the short reads, we actually can read sequence analysis, small structural variants, mitochondrial disorders, and specific STRs. With the added value of the long reads analysis, we're getting on and everything much deeper level. So from the structural variants perspective, we can get all the sizes from 50 base pairs to the entire chromosome with much better detection and capabilities, sensitivity and specificity. And that's allowing us to look better. We can actually do phasing analysis if needed to understand. We can also do with the added value of the long reads much more short tandem repeats, because we can see more genes with longer repeats, and it also allows us to do methylation. So the added value of the long reads makes it much, like, you can see the entire genome completely and reducing the limitations of the short reads.
I see. And do you have a code that is just long read only or short read only?
So we have from last year a code that is short and long reads. So the short reads only is the 0212U, and the comparator for it is the 0213U. That's for short reads only. We don't have for long reads only. We have for short and long read for singleton, and that's the 0567U.
I see. But there is no, you have not submitted a code for just long read. It is always long read on top of a traditional short read.
Exactly. Yep.
Okay. I think I see there's still a hand up. Okay, that's yours. Okay.
Yeah. Sorry, I'll take it down.
All right. So panelists, does anybody have any other questions about the service? So just to be clear, as we are discussing the service, there is traditional short read, which is what we are looking at with these crosswalk codes. So 0212U and 0213U would be a traditional short read, whole genome mitochondrial, with all of the variant classes, etc. What we are talking about here is a traditional short read with essentially a resequencing of the same specimen using a long read methodology on top of the short read methodology. Did I get that correct?
Yes.
Okay. All right. Do we have any other questions from our panelists? All right. So I think we've got all of the ballot options that we were looking for here. So panelists, please cast your votes.
Okay, thank you.
All right, so I think 74 and 75 we were okay with. Correct, because that, and because there isn't the notation of long read and short read, you're presuming this is a short read, and we've got fetal sample in there, so this is a short read prenatal genome.
Yeah, and this is the vote we conducted before as it's presented here, correct? We didn't change
Yep.
anything. Okay,
Yep.
so I think we're good on that then, yep. And then we need to talk about 77, right?
And so this one's okay.
Yep.
So just for our transcription, we're referencing 0675U is fine, or
Yes.
no revote is necessary.
Correct.
Moving on to 0671U.
Also fine, no revote necessary.
Okay.
Oh, wait, double check, go back.
Wait,
I'm sorry.
no.
No, yeah.
I think this one.
So we
I
needed
think
to,
this is a revote.
yeah, this is the one we needed to re-vote on. So
Oh.
this was similar to 73, in which the inclusion of amnio, CVS, et cetera,